Hey guys, welcome to our very first video. This video is on the nature of science and experimental design. So basically we're going to be looking at what is science, what are the aspects of science, and then how do we design a real good experiment. So first of all, what is science? Have you ever stopped to think about that? It's defined by the dictionary as a branch of knowledge or study dealing with a body of facts or truths systematically. Oh my gosh, this is boring. Basically, definitions from the dictionary can be rather drab. So let's really think about this. Let's take all the different sources that have defined science and put them together to come up with a simple, straightforward, cohesive definition of what science is exactly. So what I found is it can be broken down into three main ideas. First, science is a body of knowledge. Second, it's a set of methods and processes. And third, it's a way of knowing about the physical world. So yes, science is the facts that I'm telling you. Yes, science is the scientific method and experimentation. And it's all of that put together into a way of figuring out the world around us. It is a method of exploration, if you will. Now, what are the goals of science? So we see that it is a body of knowledge. We see that it is a process. It is a way of knowing. But why? Why do we do science? Well, we do it to understand patterns and provide explanations for the natural world. We're very curious people in general. So we want to be able to understand what's going around us, but then also we want our lives to be easier. I want you to be able to predict to me what the weather is going to be tomorrow so I can plan. We like to make predictions about natural events and understand the world around us, and that's mainly what science is for. Now, I want you to pause the video and think about this. What qualities do you think a scientist must have, or what a scientist should have? I don't want you to spend a lot of time on this, just maybe like a sentence or two listing the qualities. And when you're all done, join me in the next slide. Now, the qualities of a scientist. A scientist should be curious, right? This is at least what I think is one of the most important ones. Scientists are very curious people. They're asking all these big questions. They're wondering about the world around them. But have you ever stopped to think that a scientist must also be creative? Normally you think of scientists as being super smart people who are really good at math and they're logical like Mr. Spock. But in the end, scientists are really creative people. If you think about the fact that they are trying to answer questions that people have never even thought of before, you've got to be creative in order to be working on something like that. Scientists are also very open-minded, and what I mean by that is not that they're like, oh, I'll believe whatever comes in front of me, and oh, you say that, that's interesting, I'm open-minded. What I mean by that is they're open to possibilities. So if you look at the quote on this picture on the slide, the most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but that's funny. If you take this example, my dad is a scientist, and the lab he was working in, the people who were doing an experiment in his lab, they said, why aren't we getting the results that we're expecting? Nothing is working the way it should. It's almost as if the DNA that we're studying is being read not only this way, but also that way. And that's impossible. DNA is only read in one direction. At least that's what we thought. Scientists have to be open-minded enough to say, what if DNA can be read in two different directions? And that's actually what they discovered. Unfortunately, they were beat by another team, so they weren't the ones that got credit for the discovery. But that's still something that proves scientists have to be open-minded. They have to look at what they're studying and be open to letting the experiment carry them in new ways. And the last one, this is the one that I might say is the one thing a scientist must have. They have to be skeptical. And what I mean by that is not like that person in your family that's always like, I don't believe that, show me where Google says that's true. But what I mean more is they're constantly wondering if something can be improved or how that was done, or honestly, what the evidence is and what the data shows about that. It's more of a healthy skepticism, one where it's tied in with the curiosity. So these are what I thought were the 4 main qualities of a scientist. Maybe you had more, maybe you had less, and we can talk about that in class later if you'd like. But what are the qualities of science itself? So we call this the nature of science, aspects of science's nature. Scientific knowledge is organized into facts, hypotheses, theories, and laws. That's a big one, and we're going to discuss that more in the next couple slides. Science aims to avoid bias. We will also discuss bias a lot throughout the year. And its goal is to pursue objectivity over subjectivity. I want evidence versus opinion and feeling. Scientific knowledge is durable yet tentative. This one is kind of confusing, and we'll go over that more in one of the later slides. And just like a scientist, science itself is a creative endeavor. It's not just all logic. It's a lot of beauty and poetry in it too, if you ask me. Scientific knowledge is derived from data, which we get through observation, inference, and experimentation. This is the bulk of what we're going to focus on in our lecture today. And science is social. It's not that weird guy who's sitting in a basement lab with a white lab coat and crazy hair like he stuck his finger in a light socket. No, science is a really big community, and people are always excited to talk about it and to share their ideas and work together. Science is very collaborative. And finally, scientific knowledge is collected using a variety of tools and methods. We'll talk about that more in tomorrow's lecture, but it's essentially boiling it down to the scientific method and things like that. So how do we organize our scientific knowledge? You can have facts and hypotheses. That's how we're going to start this one. A fact is an observation about the physical world. The earth is round. The lemon is yellow. The tree is tall. Those are all facts. A hypothesis is a proposed testable explanation for a phenomenon. So if I ask the question, why is that tree taller than another one? Well, a hypothesis could say that tree is taller because it's older or it gets more nutrients. It's a proposed explanation, and it's something I can test. I can go into a lab and measure it and see how what I change affects it. Now, here's one of our ways of organizing scientific knowledge that really gets people confused. There's a lot of misunderstandings about theories and laws. A theory in science is not the same thing as a theory that you might say, oh, I have a theory about that. It's not an idea. A theory in science is something heavily supported by evidence. It's an overarching, well-tested, unifying explanation supported by many hypotheses. It has a lot of evidence to back it up. And this is what scientists use to make predictions and draw new conclusions. It's a big idea that's heavily proven that we can use to predict future events. Things like cell theory, germ theory of disease, the theory of gravity, these are all heavily supported. Now, laws are statements of fact based on repeated experimental observations. They describe a natural phenomena, so they don't explain why, they just describe it as it is. Usually this description involves math. So that's why often you'll see a lot of laws in chemistry and in physics, but not a lot of laws in biology. And something like the law of gravity, we have an actual equation that describes the law of gravity. It's very fascinating. Same with the laws of thermodynamics. But now you might say, well, how can I have a theory of gravity and a law of gravity? Well, like I said, the law of gravity is this nice equation that sums it all up. It's the statement of fact, essentially, about gravity. Whereas the theory of gravity explains how gravity works. It's the why of gravity, you could say. And that's the one that we use to predict how things will behave in other circumstances based on gravity. Now, a lot of people also have the misconception that a theory becomes a law with enough evidence, and that's not true. That's like saying something that's yellow will become something that is round. Color and shape are not the same thing. There's no way that something yellow can become something round. They're not even interconnected. So a theory is an explanation, and the law is the statement of fact. They're two different ways that we organize our knowledge. Now, earlier I said that scientific knowledge is durable, but it's tentative. So all of these theories and laws and facts and things like that, they're durable yet tentative. Durable means strong and it can withstand outside pressures. Tentative means it can change. It's a temporary thing. So how is our knowledge both? Well, the answer to both is the same thing, the scientific method. So because science focuses on evidence and a specific methodology and peer reviewer checking each other's work and data analysis and things like that, once scientists get information, it's strong. It has a lot of support to it. But we're constantly changing. We're constantly getting new technology. We're constantly finding new information. And just because I have new information, that doesn't mean my old information is bad and I have to throw it out. What it means is I have to reevaluate. So tomorrow we could find a new discovery based on a new invention, like a new telescope that helps us see something we've never seen before. Well, it might make us reevaluate what we know about the universe and space. Not throw it out, not saying, oh, well, that's not true. But we may have to say, well, what does all of that mean in light of this new information? So it's based on new evidence, better methods, better technology. And that's what's constantly keeping science changing and growing and honestly bettering itself every day. So on your paper, pause again if you need to, just like a sentence or two. Why do you think it might be a good thing that science is tentative? Or do you think science shouldn't be tentative? Do you want your science to be always right or always wrong and nothing in between? Okay, join me on the next slide when you're done. So all of this scientific knowledge that we've been talking about, how do we get it in the first place? And that's where our scientific methodologies come into play. Not just the scientific method, but our many methodologies. You probably learned in the past about that 6-step scientific method. You observe and ask a question and form a hypothesis, then perform the experiment, and it's nice and piecemeal and stepwise. But that's not how real life works. It's not quite so simple as all that. You really have a continuous process. I can make an observation and form a hypothesis, then go back out the next day and make a completely new observation that makes me reevaluate my hypothesis. I can test a hypothesis and get results that make me say, well, why did that happen? And ask a whole new question and form a whole new hypothesis. I can complete an experiment and send my results in to another scientist and they can say, that's great, but what about this other aspect of science that you didn't think about? That doesn't really fit in with what you've discovered. Or maybe what you discovered helps answer this other part of science that we've been confused about. It's a continuous living process that we move forward and backward and we jump levels. It's not quite so I do step 1, then step 2, and then move on. Now, one of the key aspects of scientific methodologies and the scientific method and all of that is experimentation. It's a procedure or a process that scientists do to make a discovery or test a hypothesis or demonstrate a known fact. This is the key thing that you think of probably when someone mentions science to you. You think of doing experiments. So this is where we're going to really focus in our information right now. How do I build a good experiment? What are the key components of a good experiment? Now, all experimentation starts with observation. It really does. You see something or you read something and you think, well, what does that tell me? what can I find out about that? So you observe, you use your senses to gather information and perceive the natural world. Then you question, and you can make an inference about that. Now, a lot of people confuse observation and inference, so let's go over those a little bit. When I observe, I'm using my five senses. When I infer, I'm drawing a conclusion based on my observations. It's not a fact. An inference is not a fact at all, but it's a foundation to form a hypothesis. So I can observe that plants only grow where water is. I can infer that plants need water to live. So the inference is going to be the foundation of my hypothesis. Let's do a little thought experiment here. You've got this picture of the footprints on the yellow background. If we're just putting our observation caps on, I can see two different types of footprints. I can see that the footprints are jumbled up here, and I can see one of the two footprints in the third panel. Those are my observations. You can take it a little bit further and say, this one seems to have one big toe in the middle. This one seems to have more rounded, tinier toes, things like that. Those are all observations. Now, an inference would be this green animal, or even that it's an animal at all. That's an inference. Who knows that it's not, two kids playing with a doll or something like that. So I am going to infer that these are animals. And I'm going to say that this animal with the green, it's walking at a steady pace. All of these are spaced fairly well equal apart. And then all of a sudden, its footsteps get further and further apart from each other. Why would that happen? Well, I'm going to infer this one started running. And then there's this big jumble over here. I'm going to infer that because the green animal started running towards the red animal and they got all jumbled up, that they fought. And then I'm going to infer that the green animal is the one that won the fight, and maybe it ate the red animal because I only see one set of footprints walking away. Another inference could be that the red animal can fly, and maybe they did fight, but it flew away. Oh, excuse me, that one, it switched slides for me. So observation and inference are very different, but overall, because of our observations, we can question, and that drives science forward. Now, I want you to think about it and write this down. I want you to give an example of an observation and an inference from your life, preferably from today, if you can really think about it. Write that down and then join me in the next slide. Now, after we make our observations and our inferences when we're experimenting, we want to form a hypothesis. So a hypothesis, as I said earlier, is a testable answer to a question or a proposed explanation for a phenomena that I'm noticing. It's a result of my questioning. It's kind of like an inference that I can do an experiment with. So hypotheses are not proven or disproven. That's another big misconception. I don't prove a hypothesis. The data that I collect can support my hypothesis or it can refute my hypothesis, one of the two. But I don't prove it one way or the other. Now, scientists might say something like plants get their necessary nitrogen from the atmosphere by taking in nitrogen gas. Now, that's not actually true. The data doesn't support that hypothesis, but that is a hypothesis. Same with an apple a day keeps the doctor away. These are things I can test. But we're not quite at the same level as other scientists yet. So what we're going to do is if, then, because statements. And these are what I'm going to call formal hypotheses. And this is just an easy way to help us really come up with a hypothesis that is testable. And it makes you think things through. So if I water a plant 3 times a day, then it will grow very tall because plants need water to live. That's the kind of format that we're going to be writing our hypotheses in for a while. Now imagine this. You're walking through the forest on a crisp October morning, obviously not a Southern Californian forest, and you're in awe at the beauty and vibrancy of the colors around you. You see green, yellow, orange, and deep, pretty reds. So you wonder, what makes leaves change color? You just started on your way to making an experiment. You made an observation and you asked a question. Now we can form a hypothesis. So maybe one person says, decreasing temperatures cause leaves to change color. Somebody else says, decreasing sunlight availability causes them to change color. And a third person says, losing water makes them change color. All of these are possible hypotheses. The difference is they're all testing what we call a different variable. Now, variables are any aspect of an experiment that can be controlled, measured, or changed. We often focus on two different variables, the independent and the dependent variable. So an independent variable is like the cause of the change. It's the variable that you control. So in an example, it would be the amount of water that I give to a plant. So I can give a plant 1 cup a day, 5 cups a day, 10 cups a day. That's the independent variable. I am changing it. The dependent variable depends on the independent variable. It's the effect. It's what you measure, the data you collect. So the amount of water I give a plant is going to change the height of the plant in the end, or how the plant grows. So the IV, independent variable, causes the dependent variable to change. That's a key statement. And if you plug anything into that sentence, you can see if it makes sense. This is a good way to test yourself on quizzes. You can insert what you think the IV and the DV is into that sentence and see if it makes sense. And we'll do examples of that in the next slide. So I change the IV. The DV depends on what I change. It's the data that I measure. So let's do some examples here. What you can do is you can either pause this and try to figure out what the IV and the DV in each of these statements are on your own and then test yourself. Or you can let me go through 1 or 2 of them and then you can pause it to do the last 2. It's completely up to you. But this is what your homework, excuse me, is going to be tonight. So let's go over this carefully together. Now, there's 2 variables in these sentences, an IV and a DV. Let's look at the first sentence. The more time spent outside causes your stress levels to decrease. So time spent outside is one variable. My stress level is another variable. Which one is the IV? Which one is the DV? Well, I can plug these into that sentence now. The IV causes the DV to change. So the time spent outside causes the stress level to change. Does that make sense? Yeah, it kind of does. But what if I said the stress level causes the time spent outside to change? That doesn't make quite as much sense. So when I spend time outside, how much time I spend outside, that is my IV, the thing that I am changing. My DV, or what I'm measuring, is my stress level. So the IV is causing my stress level to change. Now the second one, the number of hours you study will affect your score in the class. So in this one, what am I changing? What do I have control over that I am going to manipulate? Am I going to manipulate the hours I study in a class, or am I going to manipulate my score? Can you go into my grade book and change your grades? No. So the number of hours you study, that's going to be your IV. And your score in the class, what depends on how much you study, is the DV. All right, last two. We ready? The more hours you work, the more money you earn. So hours you work and money you earn. So money earned and hours worked. Which one is my IV? Which one is my DV? So is the amount of money I earn going to affect the number of hours I work? Or are the amount of hours I work going to affect how much money I get? So I am going to manipulate how many hours I work, and that's going to affect how much money I get. I don't know why it keeps doing that. DV. There we go. So the amount of hours I work causes the amount of money I earn to change. Makes sense. The last one here. The ice cream melts faster if it is very hot. So this one we're actually going to have to change a little bit. Ice cream melts faster. What's a better way of phrasing that? The time it takes the ice cream to melt versus if it is very hot. Well, that's temperature. That's what I'm measuring there. So the time it takes the ice cream to melt versus the temperature. Which one's the IV? Which one's the DV? Let's plug it into the sentence. The time it takes the ice cream to melt causes the temperature to change. That makes no sense at all. The temperature causes the time it takes the ice cream to melt to change. That one makes sense. So in this case, I kind of switched it up on you. Your IV is the temperature, and your DV is how long it takes the ice cream to melt. Okay, if you have any questions about these, write it down right now. You can pause the video and write them down and come to class with them tomorrow, so that way we can actually sit and talk about them and I can help you understand them better. All right, let's move on. Now, when we have this IV and this DV and all of our data, we like to put it into a way that we can understand and that makes sense just really quickly from looking at it. So we tend to graph it or put it in a data table. And we're going to go over these more later tomorrow in our next lecture video. But essentially, this is my x-axis and this is my y-axis. My independent variable is always on my x-axis. So this is my IV always. And my DV is always on my y-axis. I always want to have a title as well. Often it's in the form of IV versus DV, just because that really helps you understand what is going on in your graph or your table. It really helps your reader or the person looking at it to get a quick view of what they're looking at. So looking at some examples, these have titles, but people in a store. That's a pretty vague title. So if we look at what we're actually measuring here, there's number of people and then the time throughout the day. So a better title for this might be time of day versus number of people in store. Sorry, pandas, we're gonna write all over your graph. So we also notice that this has units. The time is in the hour of the day, PM or AM. We have year here, and they specify the years down below. And then you have panda population or number of people on your y-axis, meaning that these are your DVs. These are what you're going to measure. And these two down here are your IVs. This is what you are going to be changing or you're going to be looking at throughout your experiment. All right, we'll talk more about these later. Now, with our IVs and our DVs, there's certain things that change within those. We don't just have one IV is what I'm trying to say. I don't just say, I'm going to measure how fast the ice cream melts at 50 degrees Fahrenheit. What I'm probably going to do is measure it at 50, 60, 70, 80, and 90 degrees Fahrenheit. Those are levels of my independent variable. But I usually need one that I consider my baseline or my normal. So I'd probably pick room temperature to be the normal value. Otherwise, how will you know the differences? How will you know what you're comparing your data to? So let's look at the example on the screen here. So my control group for this experiment is a plant watered with just water. This one's getting water and nitrogen salt. This one's getting water and potassium, and this one's getting water and phosphorus. So I have 1, 2, 3, 4 levels of my independent variable. And I want to know what the normal or the baseline value is. That's probably going to be just water. I don't normally add potassium or phosphorus or nitrogen to my plants when I'm watering them. I just fill up a cup of water and give it to them. So I'm going to measure how tall these plants grow. So let's say the one with the nitrogen salts grows to be about this tall. And the one with potassium grows to be about this tall. And phosphorus grows to be about that tall. So that's great. You'd say, well, then give your plant nitrogen salts because they grew the tallest. But now what if our water grew to be this tall? You would say none of these are good because they all made the plant significantly shorter than the water. So you would want to just give your plant water. This is why control groups are so important. What's that normal or that baseline value? Because how do we know what's going on in our experiment unless we know what the base or the standard is? So now there's also more variables than just the IV and the DV. Now, if I were to take my ice cream outside to see how fast it's going to melt, and I take 1 scoop of Haagen-Dazs in a cone, and the next day I take 3 scoops of Baskin-Robbins in a cup, well, those are 2 different things. I can't really compare those at all. They have different variables. They have different numbers of scoops, different brands. You can look at, are they lactose free or not? One's in a cup, one's in a cone. These are all different variables that I don't want to be changing at all. I manipulate the IV and that's it. So we call these variables constants because they should be held constant throughout. If we're looking at our picture example on the PowerPoint, you have 4 drops of water with every single plant. Every single plant is getting light. I'm assuming that means the equal amount of light. Every single plant is the same species of plant. They are all in pots. One isn't outside and some are in pots or whatever. The only thing that's changing is the amount of fertilizer that they're getting. This one is getting 0 drops. This one's getting 2, 4, and 6. So I can directly say the height of the plant is affected by the fertilizer only, not because one is getting more water than the other. It essentially helps you know that what you're doing is because of your IV and not because of some other thing. So I want you to think about it. Remember, pause and write down an answer, and then when you're done, you can move on. But is doing an experiment once enough to get accurate results? If you think so, give me 1 sentence that says why. If not, what should you do instead? So to make this a little more clear, would you trust someone who came up and said, I went to the Namib Desert last year, and I spent 1 day there, and man, it was raining all day. Those deserts, they're really rainy places, aren't they? So is doing that experiment once enough to get accurate results? Why or why not? Now, let's say we did our experiment once and we got a test tube with a yellow liquid inside of it. We would say, okay, this experiment gives you a yellow liquid. But then let's say you do it 30 more times and every single time after that, you get a green liquid. Which one's the true value? Probably the green liquid because it happens so many times. This is like an outlier, or maybe even a flat out mistake that you made. Maybe you added the wrong material. This is why scientists need repeated trials, even within an experiment. This is why you don't just throw the paper airplane once. You have to throw it 3, 4, 5 times and then average those results, because you never know when some outside influence affected your results, even just once. You want to normalize your data or get it to be kind of averaged, you could say. Also, what happens if you do listen to the guy who said deserts are rainy, and you go to a desert and you bring nothing but long pants and jackets, and you don't bring sunscreen and you don't bring enough water, and you have to walk across it? Well, you're not going to really be in good shape by the end of it. So it's really important to repeat your trials. Now, all these experiments that we do collect data, and we put these data in data tables or graphs, as I said earlier, just to make them easier to understand and to see it really quick. And the data is the result of the experiment. This is the part that's our evidence. It's the direct support or the reason why your hypothesis is refuted. And once we have our data, we need to analyze it. This is where we draw conclusions. This is where we apply our data to other areas. This is really where you get to the meat and potatoes of your science. And finally, once you've done all your data analysis and you've done your experiment, you want to publish. This is the goal of any professional scientist. You want to publish your work into Nature or something like Science. Essentially what you do is you send in your research and you have graphics and your writing and things like that. And it's all about what experiment you did and what the results of that experiment were. And then scientists can talk about them and apply them to their own research and things like that. It's incredibly competitive to get published in a journal, especially ones like Nature and Science. They're kind of like the cream of the crop. And each journal has its own standards of what they accept. Science and Nature tend to have pretty high standards. They want your work to be really good. And part of that is something that we call peer review. So once a scientist presents their paper to a journal, the journal will send your paper before it gets published to a bunch of different scientists who are experts in the field. And they have to read your paper and say, this is good, this isn't. Where did you get this data from? Explain this a little bit more. This graph doesn't make sense. They're reviewing your work. And if they find out that you falsified data or if you didn't have a good experimental method, they're going to say, no, your paper doesn't get published. You have to redo everything. So this is a big reason why science can claim to be durable, our knowledge, or why science can be unbiased, because we're actually sitting there and keeping each other honest. We're double checking each other's work all the time. But there's still sources of error, and this is just a fact of life. It's not something that, you can completely get rid of. Every experiment has sources of error, like that one tube that turned out yellow when all the others were green. Some of them can be ones that you can adjust, like maybe your materials aren't calibrated properly. Maybe you're not reading it properly. Some are just flat out mistakes, like you broke a glass test tube while you were working. You got to redo that. Sometimes, though, they can have what's called bias. Now, bias is prejudice in favor of or against one thing, person, or group compared to another, often in ways considered unfair. Everyone has bias. This is, once again, one of those things that we just can't get around. It's part of being human. But scientists try to recognize their bias and try to remove it as often as possible. It's also why science is social. My bias may be different from yours, and you can catch my bias and help eliminate it. It's a really important thing to eliminate this. We don't want our opinions injected into science. Science needs to be based on evidence and data only. Can you think of examples of bias in your own life? I know one big one for me is, since I was trained as a biologist, I tend to see the world in this lens of biology. So when I'm talking with my husband about engineering, because he's an engineer, I try to put things in terms of biology, and it really makes it hard for me to understand what he's talking about. That's a bias. If I learn to remove that biology lens and just try to understand it purely from the point of the facts, I'll probably understand what he's talking about better. All right, almost there. What equipment do scientists use? Some of these you're going to have to be responsible for on a quiz. But you've got things where we measure, or we hold liquids, or we heat things up, or we weigh things, or we grow things in them. And we're going to be using a bunch of these this year. So you have beakers and Erlenmeyer flasks. These hold liquids, and we can actually measure how much is in there. We can manipulate liquids in test tubes or even solids. We have lab burners where we can heat substances up. You can hold test tubes with test tube holders because hot glass and cold glass look exactly the same. So if one of your friends was heating something up, you want to use the test tube holder or the tongs to pick up the glassware because you don't know, did they heat that up already or not? You want to wear safety goggles because we might often work with things that splash in your eyes that shouldn't be in your eyes. So that's just a way to protect yourself. We use test tube racks to put our test tubes in to keep them from rolling around. We don't use tweezers, we use forceps, but they're essentially the same thing. You have a ring stand, and you often hook it up to something like this iron ring, where this device right there hooks onto the ring stand, and then you can use your hands to do something. You can leave something inside that ring, and you can leave it alone while you go do other things, which is kind of nice. You use a graduated cylinder to measure fluids. You use these eyedroppers and pipettes to dispense liquids. So I'm going to move one liquid in a specific amount from here into another beaker. You have a burette. This thing here is kind of like a pipette, except it holds a lot of liquid, and I can use this little valve at the bottom to open or close it and get liquid dispensed into another container. I use a funnel to end up moving things from one beaker to another or one flask to another and not make a mess. Petri dishes. I put like a gelatin in there, and bacteria love that stuff and they'll grow in there for me. Hot plates heat things up. Balances will measure the mass of my objects. In biology, we'll use a scalpel to cut things up. Or we'll put things on a microscope slide and look at them under the microscope and magnify them. So these are just some of the tools that biologists use. There's so many of them that we won't even get to see or that are too expensive to even touch. So know a few of these for a quiz, mainly the ones that I've circled. Hint, hint. We'll have a quiz in a few days. All right? The last thing we're going to talk about is scientific literacy. This is really what I've been trying to get at with this whole discussion today, especially the stuff about the nature of science. So scientific literacy is the knowledge and understanding of scientific concepts and processes that are required to just live a normal life. A recent study from the University of Michigan found that only 28% of Americans are scientifically literate. And you might be thinking, so what? Math and English and reading are really what you need to know to make it in the world. Some of you may even say, yeah, it's math and English, but maybe a little bit of history, because you know history repeats itself. But think about, where did your cell phone come from? The car that you use to get to school? Or what about your house itself? Who makes sure that your house can stand up and not fall apart on itself? What about if someone commits a murder and they need DNA evidence to prove them guilty or not? What about legislation in the government that's talking about, should we give more tax dollars to the National Institute of Health to develop a new vaccine? All of these things are going to affect you one day. And that doesn't even come into the discussion of, you've been to a doctor and you're responsible for your own health. Americans are not scientifically literate, and that's a little scary right now. It's important for us to know about science, just as important as math is, or reading, or even history. So I want you, though, to tell me your opinion. Tell me what your opinion is. Do you think people need to be scientifically literate? Do you think this is important? Why or why not? Try to use at least one real life example. And remember, this is like 2 to 3 sentences max, okay? All right. So that's it, guys. That was our first video. We talked about the aspects of what science is, scientific knowledge and how we organize it, what we need to set up experiments like a hypothesis, an IV, a DV, a control, and constants. And we're going to be working on that a lot this unit. We talked about how scientists need to review and analyze their data and then peer review it and publish it, and that sources of error and bias are just part of everyday life, and we have to learn to eliminate or at least reduce those as much as we can in our scientific pursuits. I want you guys to write down any questions that you have, anything you weren't really sure of or that you're still confused about, and come to class next time prepared to ask them of me. I'll spend the first few minutes, maybe 5 to 10 minutes of class going over things. If we need to take more time than that, we will. Otherwise, I hope you enjoyed it. This one is one of the longer videos. Most of our other videos are going to be much shorter than this, okay? Hope you guys have a good night. Bye.